Now the second half of our notes for today is moving us forward so that we can be ready for a lab next class. And these notes go along with the cell cycle and mitosis. I'm going to assume that you guys do remember a lot of mitosis from freshman year before. I'll still kind of cover the major basics, but I'm going to assume that you guys remember a lot of that. However, we do have some new details in Bio 2 about the cell cycle itself and kind of the life of the cell in general. Now, the first thing I want to mention is that we are talking about this primarily to talk about cell reproduction, right? Cell division. And you want to remember that we have to be able to constantly make new cells. In order for our body to grow, develop, or then maintain itself, aka heal if you get hurt or those kind of things, we need to be able to make new cells. Now, one thing that's unique about us and all the other multicellular organisms is that we also undergo cell differentiation, which means that we make all kinds of different types of cells. And as we learned with the last part of the unit, every organelle has a function, and its structure fits that function. The same is true of your cells. Right? Every cell in your body has a particular job to do, and the structure of that cell, the shape, the size, will fit with that job. So, for instance, neurons, right? Neurons are going to be shaped a lot differently than muscle cells because they have a different job to do. Muscle cells are long and thin, but then they are full of proteins that allow it to generate force. Neurons are much longer and much thinner because they're basically like electrical wires, right? Their job is to communicate from one part of your body to another along long distances, from your brain down to your toe. So neurons are even longer and skinnier in order to do their job. Now, cells also don't live forever. So like I said, we have to also maintain the body. So we need to replace cells when we lose them. We need a way to add in new cells if we see damage or injury. Now, this is going to be really true where you are more likely to get injured or lose cells. So, for instance, your skin surface or your airway, your throat, or your digestive tract, your throat, your esophagus, your stomach, these areas constantly lose cells because mainly they run into stuff. Right? Whether we're talking about your hands rubbing up against your paper as you write, or food rubbing up against your esophagus as it moves down that tube after you swallow it, those are abrasive things, right? So you lose cells there, so we're going to constantly make new cells in the skin, in the digestive tract. Now, for a prokaryotic, very basic cell, they just split into two. Not much planning there or anything. They just turn into two cells. But for eukaryotes, for us, we have a very complex series of steps known as the cell cycle that ensures that we are prepared to appropriately divide into two new cells and that those two new cells are ready to live on their own. Now, in order to go through and actually divide, the cell has to meet a few different requirements. The first thing is that it has to accurately replicate or copy, right, its DNA, and then divide that DNA equally between the two cells that we make. And we usually call these daughter cells. Now, the second thing is that the cell also must be the correct size and have enough proteins and other nutrients to successfully turn into two cells. Right? If the cell is too small and tries to split into two, and those turn out too small, they might not survive. So the cell has to be big enough, and the cell has to have a store of proteins and things like that to divide between the two daughters. And the last thing is just bigger than proteins. The cell also has to have enough cytoplasm, has to have enough of all the organelles to divide them equally between the daughter cells. We have to have enough mitochondria, Golgi apparatus, ER, chloroplasts if we're talking about plants, so that both new cells will end up with some of those organelles. Now, the cell cycle ensures that we meet these requirements so that we make two cells that will survive and continue to grow. So here is a picture of the cell cycle. Comes from Dr. Wong's book. And I want to point out something that's super important here that we maybe don't stress as much with the freshmen. This, the blue arrow, is M phase, aka mitosis. Now, in freshman year, and certainly before that, so like in junior high, we really stress for students to learn mitosis, right? Prophase, metaphase, anaphase, telophase. And a lot of times I think that we stress that so much that we lose sight of the fact that mitosis is the smallest part of a cell's life, right? All the rest of this circle, the other, 85, 90%, if this is a pie graph, right, or something, is interphase. And so by far, the longest and biggest part of the cell's life is interphase. Right, the most time spent is going to be spent in interphase. An interphase has 3 stages itself that we'll talk about here in a minute. And then you have mitosis. Now, the other thing on this picture that we will come back to in a minute is notice these check marks. There are checkpoints in the cell cycle where the cell makes sure that everything is good to go before it moves forward. And that is part of how the cell controls its cell cycle, which is also extremely important. Alright, so first let's talk about interphase. So again, remember, interphase is the longest part of the cell cycle, with mitosis just making up a little bit of it. And interphase is actually broken into 3 pieces. You've got G1, S, and G2. Now the G stages stands for growth, so that's the main thing that's going on there. G1, or the first growth stage, is where the cell grows, makes new RNA, makes new protein. It's the everyday life of the cell. Notice G1 comes right after we finish mitosis. So, in other words, when a new cell is made or a new cell is born, it moves into G1. So there's a lot of growing and developing that needs to take place there. Now, something that's kind of interesting is that during the G1 phase, you have a really important checkpoint. And that checkpoint will show up in these notes later in a minute. We call it the restriction point. The reason we call it the restriction point is because if the cell passes this G1 checkpoint, it will either divide or it will die trying. So if the cell goes through the G1 checkpoint, it is fully committed to cell division, and it will either successfully divide into 2 new cells or it will die in its attempt to turn into 2 new cells. Now, if the cell does not pass the checkpoint, it's no big deal. Right? If the cell does not pass the checkpoint, it's totally fine, and it just moves instead into what we call G0 stage. G0 sometimes is referred to as a dormancy of the cell, but I don't like that, because when we say something's dormant, it means it's like asleep, it's not doing anything, and that's not true. G0 is where the cell is doing all of its normal functions, but it is just not actively dividing. Right? No longer dividing. So a G0 cell is not actively dividing, but it is totally doing what it is supposed to be doing. So, the majority of cells in your body are actually in G0 stage. Your pancreatic cells that are making insulin, they're in G0. You don't have any reason for them to be dividing right now. So they're in G0 doing their job. Your liver cells, G0. Your bone cells, because most of you guys probably won't get much taller, G0. They're doing what they're supposed to do. They're just not dividing right now. Now, if we need them to, a lot of times we can call them back out of G0. Right? We can make them actively divide if we do have damage to an organ or something like that. But that's a discussion for another day. Now, if the cell doesn't go to G0, so if the cell does pass the checkpoint, so now I'll put a little check mark, okay, we did pass the checkpoint, then it will move on into S stage. And you got to know that S stands for synthesis of DNA. Right? This is where we copy our DNA so that each new cell, after cell division, will each get a copy of the DNA. Now, once that DNA is correctly copied, we move into G2. The second growth stage. And this is a lot like G1, except now this comes right before cell division. So now during G2, the cell will be dividing, the cell will be growing, but it will be specifically preparing for division. So it's almost like now it is getting ready, it's packing its bags because it knows a trip is coming, right? It is making sure everything is in order so that it can actually do cell division. And after we finish G2, we move out of interphase completely into mitosis. And after mitosis, we end up with two identical daughter cells in the end. Now, mitosis. Mitosis should be familiar to you guys. You've definitely had it in a couple of classes in the past. Mitosis has 4 basic stages. You can remember PMAT, right? Prophase, metaphase, anaphase, and telophase. Let's look at each one individually here. Okay, step 1 is prophase. Prophase is the most complicated step. So what I always teach students is, you remember what happens in all the other stages, and then just know that everything else happened back in prophase. Alright, so during prophase, the nuclear membrane will break down, so the nucleus goes away. The DNA condenses and changes from being this like tangled little string to being actual visible chromosomes, which you see sort of in purple here in this picture up here. Now, each chromosome is actually a pair of sister chromatids put together to form sort of an X-like structure and held together at a central node called the centromere. So centromere is my little circle here. The sister chromatids would be each half of this X, the blue and the pink. Now, meanwhile, in the cell, we start to develop a mitotic spindle, right? A football-shaped grouping of microtubules that are anchored at either end by an organelle or organelles called the centrioles. The last thing that happens then in prophase is a part of the centromere, referred to as the kinetochore, will connect or attach each pair of chromatids to this mitotic spindle. So we're going to end up with our little X's attached to our mitotic spindle. Now, a lot of times students get confused here between centromere and kinetochore. Kinetochore is just sort of like this little claw or something like that, clasp, that's part of the centromere that can grab a hold of these microtubules and move the chromosome. So again, a lot going on during prophase. The other ones are way simpler. Metaphase. Metaphase comes next. When you hear metaphase, think midline. Right? This is when the sister chromatids, the little chromosome pairs, move and line up down the middle of the cell. And when we see this, we call it the metaphase plate. Metaphase plate, when the chromosomes line up along the midline. Next step, anaphase. Remember, apart. Right. Metaphase, midline, anaphase, apart. Now, the sister chromatids will be pulled apart, one copy of each pair moving to the opposite end of the cell, or the opposite poles of the cell. This makes sure that each new daughter cell we make gets its own copy of the DNA. So, in that last slide, I drew something like this for the chromosomes. Now what will happen is the pink half will move to one side, the blue half will move to the other. And that is anaphase, pulling them apart. Now, when our two sets of chromatids reach the poles of the cell, or opposite ends of the cell, we have reached the phase of telophase. Now, in telophase, the chromosomes get to the ends, the nuclear membrane redevelops, so we end up with 2 nuclei in our cell here. DNA starts to kind of expand and become less visible, become grainy, and just this little tangled string. And that is then followed by cytokinesis, cell splitting, where the cytoplasm, as you can start to see here in this one, will divide into 2, and we'll end up with 2 separate daughter cells. Once we've completed cytokinesis, these cells move into interphase, and they go into that first part of interphase, G1. Now, something else to point out right here. Notice that mitosis itself, prophase, metaphase, anaphase, telophase, is really just nuclear division. And then that nuclear division of the chromosomes is followed by cytokinesis, a division of the cytoplasm. Now, both of these daughter cells we make are going to be identical to each other and are going to have they are going to have the same exact set of DNA. Now, the last thing we have to cover, I know this has been a lot to listen through, is that you also have to control or regulate the cell cycle. Cell growth or cell division that goes out of control is a really, really bad thing. And the reason it's a really, really bad thing is because that's what we call cancer. So your cell has a very complicated way of maintaining its control over the cell cycle. Now, we're only going to brush the surface of how that works, and I think it's still going to be a little bit confusing. But just realize that it is very complicated and complex, and that's a good thing because actually it keeps your cells in line in most situations. Now, there's several different layers of control. The first thing is that we have all those checkpoints that we saw in that picture before. Checkpoints where the cell makes sure that everything is ready to move forward before it is allowed to move forward. So the first one we talked about was that G1 checkpoint called the restriction point. Once the cell gets the okay at the restriction point, it is committed to dividing, which again means that if it doesn't successfully divide here, the cell will actually kill itself. So there's no going back once we pass this restriction point between G1 and S. Now, we then have other checkpoints as well, where we seek out problems with the cell. And if a problem is recognized, the cell will stop the cell cycle, and special proteins will try to fix the problems that are there. So you got proteins that check for issues with the DNA, or issues with not enough cell size, or not enough organelles. And if there's a problem, the cell just freezes, right? It goes into cell cycle arrest. So the immediate reaction isn't just to die or something, but the immediate reaction is, let's hold on for a second, and let's see if we can fix this. It's kind of like if you think of something that's made in a factory, like a car, right? It has this production line, and it moves down this line, and different people are supposed to put this bolt here, or screw something on here, or whatever. If somebody notices an issue, they can stop that production line, right? Stop the conveyor belt for a second, fix what's going on, and then once it's fixed, let it go again. That's what the cell does. It freezes the cell cycle for a moment, tries to fix things, and then pushes it forward. If the cell cannot be fixed, though, then the cell is killed. Now, that sounds a little brutal, but it's the way that the cell can avoid becoming cancerous or dividing out of control. And it will kill itself very quietly in a process called apoptosis, one of my favorite words in biology. Apoptosis, controlled cell death. The cell will quietly kill itself, not affecting all the cells around it, but basically just removing itself from the population to ensure that it will not become diseased, or a term that we really already know, even though we haven't talked about it in class much, or it avoids becoming cancerous. Now, I do want you to also know the alternative to apoptosis, which is necrosis. Now, necrosis is massive cell death. You guys could probably give me a couple of reasons why we might see necrosis. The major ones are bacterial infection, like really bad bacterial infection. Another one that sometimes people bring up is toxins, like spider bite, something like that, can cause just this massive cell death. Now, if you get a really bad, like a brown recluse spider bite, or a really nasty bacterial infection really on someone who has really bad hygiene, somebody who has not cleaned themselves at all, you can get a really nasty infection that will start to cause so much cell and tissue death that the cell will like start to, or the skin will like start to rot away. I saw a case of this with an individual who, not to judge here, but who was homeless and did not take care of himself and was in a hospital. He had such bad bacterial infection on his skin that it had started to really turn almost blackish green, and it was just like becoming goo in this one area of his leg. And it smelled so bad. It stinks terribly when you see this necrosis. So, in other words, long story short here, necrosis is bad. We're not supposed to see that, right? But to avoid any kind of massive cell death, we have apoptosis, carefully removing one cell. Now, I don't want to be insensitive or anything like that, but there's a difference here that a college professor pointed out to me, and I think it's a great way to think of these two things. Apoptosis is if someone kills themselves quietly in their home, not affecting anyone else when they do so, other than obviously the psychological issues of family and friends and things. But that one person doesn't hurt anybody else when they do that. Necrosis is quite the opposite. Necrosis is like someone who decides that they want to kill themselves, so they strap some explosives to their body and go to the most populated area they can find, and they blow up 500 people. Because apoptosis causes the death of one cell and doesn't harm the ones around it. Necrosis, one cell essentially bursts, and all of its internal kind of components cause cell death around them as well. So, the body needs to be able to do apoptosis, but it always wants to avoid necrosis. Now, the last thing we have here is a little detail about how these checkpoints work. Now again, this is just barely skating the surface of this. There's a lot of detail involved here. But I want you to recognize two different types of protein that are involved in these checkpoint controls. And they're called cyclins and CDKs. Now cyclins are named very well because cyclins cycle, right? The amount of cyclins, or the concentration of cyclins, fluctuates throughout the cell cycle. So you see the concentration increase and come back down, and increase and come back down. And that's what this bottom picture is showing you. I know it's small, is a couple of different types of cyclin molecules and how they might fluctuate. So you see the yellow line here, it kind of goes up and down, and the green and the pink and the blue one have spikes where they go real high and then they go away completely. Cyclins fluctuate throughout the cycle, but they interact with another protein called a CDK, or a cyclin-dependent kinase. Now, a kinase is a group of enzymes that activate stuff. Okay? But the reason CDKs are unique is CDKs exist at a constant concentration at all times. So, I'm going to kind of add to my picture up here. So the black line is cyclins, and it says it's sort of like a graph. The blue line is CDKs. Now here's why it's unique. CDKs move the cell forward in the cell cycle. So CDK's job, their function, is to push the cell through checkpoints, push the cell forward. But CDKs only work when there's enough cyclin to activate the CDK. So the way the cell does this is that if the checkpoint should be passed, if the cell is ready to divide, it will increase its cyclin concentration, which activates CDKs and causes the cell to push forward through cell division. If the cell is not ready to divide, it will dampen its concentration of CDKs so that they do not interact, it will dampen its concentration of cyclins so that they will not interact with the CDKs, and you will not get crossing of the checkpoint and movement through the cell cycle, and therefore not cell division. So it's kind of tricky. We can talk about it more in class if we need to. But the interaction of these two proteins together is the way that we control the cell cycle. Okay, I know that was a lot to go through, but it gets us to a really good point, so we can do a lab next class. We can keep moving forward. Thanks for listening. Hope you've had a good fall break. Take care.